Dear student, I am Dr. 𝗠�‌𝐲𝐿𝐫𝐞, a professor in biochemistry. I have done MD in biochemistry from 𝗏𝐻𝐶𝑢𝑟𝑏A Medical College, Manipal University. In this video, I will be explaining you all about folic acid. Now, the folic acid is also referred as vitamin B9. Now, the folic acid, it is rich in green leafy vegetables and hence the name is folic acid, because in Latin folium means leaf. So, since it is available richly in green leafy vegetables. Now, folic acid, it is a combination of a pteridine ring, para-aminobenzoic acid and glutamic acid. This goes in sequence like this: pteridine ring, para-aminobenzoic acid, glutamic acid. Together we refer it as folate or folic acid. Folate is a deprotonated form of folic acid. And this folic acid, it plays a key role in the transfer of 1 carbon units in several biosynthetic processes in our body. And what are the other molecules other than folic acid that are involved in 1 carbon metabolism? It is biotin, which is involved in carboxylation reaction, and 𝗏𝐻-adenosyl-methionine, which is acting as a methyl group donor in some of the reaction. So, I am writing down now. So, 1 carbon metabolism, donors of carbon atoms in 1 carbon metabolism. It is a Tetrahydrofolate derivative, which is actually the active form of folic acid. Second is biotin dependent carboxylation process, and also other carboxylation which are independent of biotin, basically the carboxylation. Number 3 is 𝗏𝐻-adenosyl-methionine, which is also acting as a 1 carbon donor there. Now, let's move on to see the synthesis of folic acid, structure and synthesis of folic acid. Structure I have already explained you. It is a pteridine ring which is attached to para-aminobenzoic acid with the glutamate. Now, how that is synthesized? Pteridine ring, which is condensing with para-aminobenzoic acid with the covalent linkase, and this will be created by enzyme steroyl synthetase. This steroyl synthetase enzyme, it is going to condense pteridine ring with para-aminobenzoic acid to make 𝗏𝐴𝐸𝐼c acid. And once we get 𝗏𝐴𝐸𝐼c acid, this 𝗏𝐴𝐸𝐼c acid, it will be attached with glutamate to make folate. Okay. So, once we get folate, this folate, it has got it is now attached with the 1 glutamate molecule. Folate is attached with 1 glutamate, and that glutamate is attached with 5 more glutamate molecule. Basically, a glutamate which is attached to a folate in the basic structure of folic acid, which is pteridine with para-aminobenzoic acid making 𝗏𝐴𝐸𝐼c acid. 𝗏𝐴𝐸𝐼c acid combined with glutamate to make folate, and to that folate additional 5 glutamates will be added. Okay, so first glutamate in the folate and 5 more glutamates will be added to our original glutamate molecule to make a polyglutamate tail, and that polyglutamate tail, it imparts multiple negative charges to a folic acid, and that will basically make this folic acid to be sequestered within the cell where this folic acid is required. Now, what happens to the folate that is made in the cell? Because the first 2 reactions that we are seeing here in this particular slide will be going on in the microbials or microbial organisms or bacteria. These 2 reactions will be going on in the bacteria, steroyl synthetase and formation of folate. And in both microbial organisms like prokaryotes and also in eukaryote, the folate will undergo reduction process. It is a double reduction going on. folate will be converted into tetrahydrofolate, Thus, that will be done by enzyme dihydrofolate reductase, referred as DHFR. Okay, so this is how tetrahydrofolate molecule is synthesized, both in the bacteria and in eukaryotes and in the humans. Okay, so in the bacteria, pteridine combining with para-aminobenzoic acid to make toroic acid, that will be done by enzyme toroyl synthetase. Once we get toroic acid, it will be added with glutamate to make folate, and to this folate, especially in the plants, what happens is folate is attached with 5 more glutamate molecule to make polyglutamate folate. And then once we consume the plant source which is rich in folate, so in our intestine, that polyglutamate tail of folate will be separated into monoglutamate form and we absorb that. And once it gets into our body, it will be converted into tetrahydrofolate, and that will be done by dihydrofolate reductase. Now, as you see here in this particular figure, that there are certain pharmacological agents that will act on some of the reactions that are going on here. That is, toroyl synthetase enzyme is inhibited by sulfonamides, thereby decreasing folic acid synthesis in the bacteria. Dihydrofolate reductase enzyme is inhibited by trimethoprim, methotrexate, aminopterin, amithopterin. All these are anti-cancer drug or anti-tumor drugs which will act on DHFR, thereby decreases the conversion of folate into tetrahydrofolate molecule. Now let's see sequential activation of folic acid. Sequential activation of folic acid, it will be done by a single enzyme, that is dihydrofolate reductase. And this dihydrofolate reductase, what it does, it is going to add 2 protons initially to folate to make it as dihydrofolate, that is written as dihydrofolate there. So the DHFR is the enzyme, dihydrofolate reductase enzyme. And now this DHF, that is dihydrofolate, is further added with 2 more proton to make it as tetrahydrofolate. And the proton donor here is the NADH plus H plus molecule. Now, once we have tetrahydrofolate, and this tetrahydrofolate is an active form of folic acid, and this is involved in 1-carbon metabolism. So there are different types of 1-carbon derivatives, all of them go into variety of reactions in our cell. We are going to look into that as we move on. Now, before we get into 1-carbon metabolism in detail, let me explain you some of the folate antagonists, some of the drugs that inhibit synthesis of folic acid, both in the bacteria and also in humans. So, anti-tumor drugs, that is methotrexate, amithopterin, aminopterin, all of them, so they competitively inhibit dihydrofolate reductase enzyme, and thereby they decrease the availability of tetrahydrofolate, and when the tetrahydrofolate levels are less in the cell, so that means 1-carbon metabolism do not go on. And one of the important function of 1-carbon metabolism, a carbon donated by tetrahydrofolate, is the synthesis of purines and pyrimidines, and also in the conversion of deoxyuridine monophosphate into deoxythymidine monophosphate in DNA synthesis. So because of the decreased availability of tetrahydrofolate, so DNA synthesis will be decreased, thereby proliferation of cells will be decreased, and that will decrease the tumor size. And this is how anti-tumor agents like methotrexate, aminopterin, amithopterin, all this will work. Now, there is something called as leucovorin rescue. And what is this leucovorin rescue? There is a leucovorin agent called N5-formyl tetrahydrofolate. Let me write it down. N5-formyl THF, tetrahydrofolate. And this N5-formyl tetrahydrofolate is also called as folinic acid. And what is the mechanism here? What is leucovorin rescue? So, leucovorin N5-formyl tetrahydrofolate, it is used as an antidote for cells that have decreased levels of folic acid during methotrexate treatment. Whenever we give methotrexate as a treatment for leukemia, so this methotrexate is taken up by all the proliferating cells at higher concentration. Some of the normal cells may also take the methotrexate, so thereby they will decrease their DNA synthesis, cell proliferation can be decreased. That's the mechanism of methotrexate. But thing is, we want to target only the tumor cells. We don't want to affect the normal cells. We don't want to decrease the dividing capacity of the normal cells. How to avoid that? So, the answer to that is providing N5-formyl tetrahydrofolate. And this N5-formyl tetrahydrofolate given 24 hours after giving methotrexate, thereby N5-formyl tetrahydrofolate will be taken up by the normal cells where they can resynthesize their 1-carbon metabolites and normalize their cell proliferation, whereas tumor cells which are taken high concentration of methotrexate are affected, thereby they won't be able to synthesize DNA properly and the proliferation of the cells will decrease. Now, there are certain drugs called PABA analogs. This PABA analog, example for PABA analog is sulfonamides. And sulfonamides, they are going to decrease or they are going to inhibit pteryl synthetase enzyme, as we have seen in the previous figure. Para-aminobenzoic acid analogs, sulfonamides, they decrease condensation of para-aminobenzoic acid with the pteridine ring to make pteroic acid, right? So, that particular reaction is inhibited by sulfonamides, especially in the microorganisms, thereby folic acid synthesis in them will be inhibited, thereby decreases the proliferation of the bacteria and thereby decreasing the severity of the infection. That's how sulfonamides will work. Coming to trimethoprim. This trimethoprim, it inhibits dihydrofolate reductase enzyme, thereby decreases tetrahydrofolate formation. So, that's why trimethoprim is used synergistically with sulfonamides. It basically affecting two sequential reactions and making the drug most effective against the bacteria. First thing is sulfonamides going to inhibit pteryl synthetase, which is affecting the conversion of para-aminobenzoic acid condensation with pteridine ring to make folic acid, and folic acid conversion into tetrahydrofolic acid, which is done by DHFR, is also blocked by trimethoprim. So, that means trimethoprim sulfonamide combination will act on two important reactions in tetrahydrofolate formation, thereby making a drug most effective against bacteria. So, that's how these drugs will work. Now, let's move on to see absorption and distribution of folate in our body. Folic acid is mainly absorbed in the small intestine, especially in jejunum. And folate, which is coming into our body, coming from plant source as polyglutamate folate form. And since this polyglutamate has got like multiple glutamate, at least like 6 glutamate molecules are there attached. And these glutamates, as you know, glutamate is a negatively charged amino acid. It carries multiple negative charges, and that makes it less efficient for absorption by the intestinal epithelial cells. So, this multiple glutamates need to be removed, like at least 5 glutamate molecules need to be removed from the polyglutamate to convert that into monoglutamate form. And the conversion of polyglutamate into monoglutamate form, it will be done by enzyme gamma glutamyl hydrolase. That is gamma glutamyl hydrolase enzyme. Gamma glutamyl hydrolase enzyme is the one that will convert polyglutamate into monoglutamate form of folate. Now, this monoglutamate form of folate is absorbed and it will be converted to N5-methyl tetrahydrofolate. Make sure you remember this molecule. So, the major circulatory form of folic acid in our body is N5-methyl tetrahydrofolate, and this is the molecule which is entering into the portal blood, and this is the major circulating form of folic acid in our body. And in the blood, like in the portal blood, N5-methyl tetrahydrofolate, it is going to go and bind with two globulins and transport it in the blood. So from the blood, the folate is taken up by tissues and they are going to be used for metabolic functions, and some of the folate is specially taken up by the liver, and liver is going to store this folate as N5-methyl tetrahydrofolate, and this is a storage form of folic acid. So both a circulatory form of folic acid and also storage form of folic acid is N5-methyl tetrahydrofolate. Make sure you remember that. And this N5-methyl tetrahydrofolate, once released by the liver, it can be converted into various other coenzyme forms. There are different forms of folate derivatives. We are going to see that as we move on. Now, since I told you liver is the organ where it absorbs most of this folate and it is going to store it as N5-methyl tetrahydrofolate. So the liver contains about half of the total body stores of folate. Because of this supplemental liver storage, folates tend not to drop as quickly as other water-soluble vitamins, with exception to vitamin B12. So that means folate is one of the water-soluble vitamin that is significantly stored in our body, and the amount of folate that is stored in our body is sufficient to meet the needs of our folate for 3 to 4 months of dietary folate deficiency. So that means when there is a dietary folate deficiency, from that point onwards, it takes 3 to 4 months for folate deficiency manifestation, signs and symptoms to appear. Now let's move on to see what are the different coenzyme forms of folic acid. Now the active form of folic acid is tetrahydrofolate, written as THF, and this tetrahydrofolate can have a different 1 carbon units, and those 1 carbon units can be methyl group, that is CH3, or it can be methanyl group, as it is written here, methylene group. Methylene group is CH2, or it can be methynyl can be triple bond CH, or it can be formyl, that is CHO, or it can be CHNH, that is formimino. So these are different forms of tetrahydrofolate derivative. One more time. So we have methyl derivative, we have methylene derivative, that is CH2 attached to tetrahydrofolate. We have methanyl derivative, CH attached to tetrahydrofolate. We have formyl derivative, we have formamino derivative. And these 1 carbon units are bound to one or both the nitrogens present in the tetrahydrofolate molecule, especially nitrogen that is present in 5th carbon or the 10th carbon of folic acid will accept these derivatives. Can be like methyl group, methylene group, methanyl group, formyl group or formamino group. All of them, either they will bind to 5th carbon or they will bind to 10th carbon in a folic acid. Now coming to folic acid that will act as a 1 carbon donor or 1 carbon acceptor. Tetrahydrofolate accepts 1 carbon unit in catabolic reactions, or it will transfer 1 carbon unit to a specific acceptor in an anabolic reaction. So the figure here will show you, so the catabolic reactions where 1 carbon units are transferred, or the anabolic reactions where 1 carbon units are donated. So basically the carbon 1 donor are shown in the left side of this figure, and the C1 acceptor, 1 carbon acceptors on the right side of the figure. As you see here, so the tetrahydrofolate is converted into 1 carbon derivative of tetrahydrofolate, and the 1 carbon donors here can be amino acid serine, glycine, histidine. Those are all 1 carbon donors to make tetrahydrofolate into a carbon derivative of 1 carbon derivative of tetrahydrofolate. And this 1 carbon derivative of tetrahydrofolate can donate that 1 carbon, that is a single carbon unit, to molecules that are involved in synthesis process. What are the molecules that can be synthesized? It is the purine synthesis, it is methionine and choline formation, or it can be formyl methonyl tRNA involved in prokaryotes, protein synthesis, thymidylate formation in DNA synthesis, and serine synthesis. Let's look into each one of these reactions in detail. Now, donors of 1 carbon units to tetrahydrofolate. The first one is the conversion of serine to glycine, and this will be done by enzyme serine hydroxymethyl transferase, where tetrahydrofolate, it is going to receive a 1 carbon unit from serine to make N5N10 methylene tetrahydrofolate. In this process, there is formation of glycine, and this is how serine is converted into glycine molecule. Now, the second reaction where glycine breakdown happens by a glycine cleavage enzyme, and in this process, glycine, when it is broken down into carbon dioxide and ammonia, so the carbon of glycine, it is given to tetrahydrofolate to make N5N10 methylene tetrahydrofolate. This is how N5N10 methylene tetrahydrofolate is formed. Basically, the breakdown of glycine, deamination of glycine here is going to give N5N10 tetrahydrofolate, because the carbon, that is the alpha carbon of glycine, is the one that is attached here to tetrahydrofolate as N5N10 methylene tetrahydrofolate. And the breakdown of histidine, when histidine breaks, it is going to convert into 1 carbon unit from form-amino-glutamic acid, and that will be transferred to tetrahydrofolate. So, this Fig loop, form-amino-glutamic acid, which is the intermediate in the breakdown of histidine, is the one that will transfer its 1 carbon to tetrahydrofolate to make N5 form-amino tetrahydrofolate, and glutamate molecule will be released. So, like this, 3 reactions here which will act as a donor of 1 carbons to tetrahydrofolate, as you see here. All these number 1, number 2 and number 3. 1, 2 and 3. These are the 3 reactions. First is serine conversion into glycine. Second is breakdown of glycine. And the third is histidine breakdown. So, when this happens, there will be formation of different 1 carbon units. So, one is N5N10 methylene tetrahydrofolate, that's 1 carbon unit. Second, we have seen another N5N10 methylene tetrahydrofolate, and the third one we have seen formation of N5 formyl tetrahydrofolate. So, these are the 1 carbon derivatives that are made upon the conversion of glycine to serine, upon breakdown of glycine into ammonium ion, and upon breakdown of histidine. Okay. Now, let us move on to see what are the acceptors of these 1 carbon units. So, first thing is purine nucleotide biosynthesis. So, in the purine nucleotide biosynthesis, so there is a carbon number 2 and carbon number 8. So, C2 and C8 of purine ring are derived from N10 formyl tetrahydrofolate. And now the second acceptor of 1 carbon derivative is conversion of homocysteine into methionine. In the conversion of homocysteine into methionine, a 1 carbon derivative that provides that 1 carbon unit is N5 methyl tetrahydrofolate, which is involved in the synthesis of methionine from homocysteine. Let me write it down. It is when homocysteine is converted into methionine. In this process, N5-methyl THF, N5-methyl tetrahydrofolate, is going to give its 1 carbon unit and convert it into just tetrahydrofolate, and that means that methyl group will be donated to homocysteine to make methionine. So this is a methyl transfer process. Methyl group is transferred from N5-methyl tetrahydrofolate to homocysteine to make it as methionine, and in the process tetrahydrofolate will be simply released. Okay, now that's where 1 carbon unit is transferred. Now the synthesis of formyl methynyl tRNA, which is required for initiation of protein synthesis in prokaryotes, especially in the mitochondria, and then the methylation of deoxyuridylate to deoxythymidylate, basically in the conversion of DUMP into DTMP done by thymidylate synthase enzyme, and during this process there will be use of N5, N10 methylene tetrahydrofolate. N5, N10 methylene tetrahydrofolate is used in the conversion of DUMP to DTMP, that is done by enzyme thymidylate synthase enzyme. Now, coming to synthesis of serine. So, in the synthesis of serine also, where there is use of 1 carbon derivative, where glycine is converted into a serine molecule. So, that will be hydroxymethyl group of serine will be required for the formation of glycine. So, basically interconversion of glycine and serine is mediated by N5-N10 methylene tetrahydrofolate. Now, synthesis of choline. In the synthesis of choline, where serine to choline conversion requires methyl group which is donated by N5-N10 methylene tetrahydrofolate. So, basically there are 3 reactions that I have explained here. All 3 of them needs this N5-N10 methylene tetrahydrofolate, that is in the conversion of DUMP into DTMP, in the synthesis of serine, and in the synthesis of choline. In all 3 of these reactions, there is a requirement of N5-N10 methylene tetrahydrofolate. Now, let us move on to see what are the causes for folate deficiency. One of the important causes for folate deficiency is whenever there is increased demand for folic acid, especially during pregnancy. So, it can lead, if sufficient supplementation is not done, it can lead to folic acid deficiency, or it can be poor intestinal absorption of folic acid, maybe because of defect in the intestinal epithelial cells or some of the disorders of intestine which are decreasing the absorption of folate. And chronic alcoholism. Alcohol, as you know, it is an anti-vitamin. That means it decreases the absorption and transport of folic acid here. And overcooking of green leafy vegetables is one of the important cause for folic acid deficiency, because green leafy vegetables are rich source of folic acid. Whenever green leafy vegetables are overcooked, basically pressure cooking of green leafy vegetables will decrease the active folic acid, thereby put a person at risk of folic acid deficiency. And also prolonged use of folate antagonist like methotrexate. Prolonged use of methotrexate may also put a person at risk of folic acid deficiency. Not just methotrexate, it can be aminopterin, amitriptyline, trimethoprim. All these agents, whenever used for a long period of time, that will put a person into folic acid deficiency. Now, let's look at effects of folic acid deficiency. Now, let's look at what are the effects of folic acid deficiency. Now, the folic acid, it is necessary for DNA replication and cell division. The reason for that is folic acid is necessary for synthesis of purines, that is carbon 2 and carbon 8 of purines, purine bases, especially adenine and guanine, these are the purine bases. So, the carbon 2 and carbon 8 of these 2 purine bases are coming from N10 formyl tetrahydrofolate. So, folic acid deficiency means N10 formyl tetrahydrofolate do not come in, that means purine bases are not synthesized. And the second one is the pyrimidine deoxythymidine monophosphate synthesis needs N5-N10 methylene tetrahydrofolate. So that is also not coming in folic acid deficiency. So overall, purine and pyrimidine availability is less. That means DNA synthesis, that is replication, DNA replication is less here, and that will affect cell division, so cell proliferation. So that's how folic acid deficiency affect cell proliferation. So because of this, what will happen? Especially in the red blood cell. So when the red blood cell is going through the developmental phase, like erythroblast into erythrocyte. So in the erythroblast, red blood cell has got nucleus, and that nucleus has to make mRNA and protein synthesis has to go on. So that process will be affected if there is a folic acid deficiency. That means nuclear size will be very small, cytoplasm will be too much. So there will be nucleocytoplasmic disproportion, and that makes a red blood cell big. So that will lead to megaloblastic red blood cell, and that is what is nucleocytoplasmic disproportion leading to megaloblast. And these megaloblasts are they can be removed from the circulation by the spleen, and thereby it can lead to hemolysis and decrease in the hemoglobin level. And the cells which are most vulnerable to deficiency states in folic acid is the rapidly dividing cells of the bone marrow and the intestinal mucosa, as they are most affected by slower than normal rates of cell division, because purine and pyrimidine biosynthesis is not available here, DNA replication is not going on sufficiently. That's why rapidly dividing cells are affected. Now, because of this, what are the consequences? What are the clinical features of folic acid deficiency? You can put together it as megaloblastic anemia. Folic acid deficiency will give rise to megaloblastic anemia. What are the changes that you are going to see in megaloblastic anemia? So, patients will show macrocytosis. Macrocytosis. What is macrocytosis? Macrocytosis means the red blood cells are big in size and they are in blastic stage. Okay, and you can put it as macroovulocytes. And the presence of macroovulocyte is indication for folic acid deficiency, which is one of the important signs that we need to look for. Now, formation of other blood cells like platelets, granulocytes are also compromised other than the red blood cells. And when you look at peripheral blood smear, so the peripheral blood smear will show something called shown as macroovulocytosis, and patients will show teardrop cells and hypersegmented neutrophils. Now, if you look at macrocytosis, the RBC size are usually larger than normal. Macrocytic oval shaped, like egg shaped macroovulocytes is the diagnostic feature of folic acid deficiency and also cobalamin deficiency. We are going to see that. And also most macrocytes lack the central pallor because of lack of hemoglobin there. And also patients will show in the peripheral smear, you are going to see anisopoikilocytosis. Anisopoikilocytosis means there will be variation in the size and shape of red blood cells. So what kind of variation we are going to see? So we are going to see like ovulocytes, teardrop cells and fragmented cells are usually found in the peripheral smear. And also patient in the peripheral smear we can see inclusion bodies. What kind of inclusion bodies? Howell jelly bodies, Cabot rings, basophilic stippling may be seen. Coming to white blood cells, in the white blood cells, white blood cell count can be decreased, and one of the most important point that you need to look for in the peripheral smear is hypersegmented neutrophils. And how many segments are generally seen here? There are at least 5 lobed neutrophils are seen per 100 ml, 100 white blood cells. Like out of 100 white blood cell, 5 white neutrophils showing lobes. 5 lobed neutrophils there. Or at least 1 6-lobed neutrophil, if it is seen, that is diagnostic of hypersegmented neutrophil, which is commonly seen in folic acid deficiency, and also it will seen in cobalamin deficiency. Coming to platelets, the platelet count is usually reduced. Giant platelets may also be observed. Reticulocyte count can be normal or it can be low because of the hemolysis that is going on there. Alright, now let's move on to see what are the pregnancy related complications that are seen in folic acid deficiency. Now, the pregnancy related complications in folic acid deficiency, one of the thing is infertility. Infertility is common both in males and females in folic acid deficiency, and also it can be seen in cobalamin deficiency. So, the infertility in men and women can be because of folate and cobalamin deficiency. Now, the maternal folate deficiency has been implicated in prematurity, and because of that there can be frequent fetal loss, and also the newborns or fetus can show neural tube defect. So, the neural tube defect is seen in both in folic acid deficiency and also in cobalamin deficiency. So, both folate and cobalamin deficiency have been implicated in frequent fetal loss and neural tube defects. Up to 50% of cases of neural tube defects are due to folate deficiency during pregnancy, and 400 to 800 micrograms of folate is recommended for those who are planning a pregnancy. That means prenatal folate levels need to be normalized, and also in the very first trimester of pregnancy, folate supplementation has to be given so that we avoid neural tube defects. Conditions that may exacerbate folate deficiency may include malabsorptive disorders and use of certain drugs like phenytoin, sulfonamides, methotrexate, which will decrease folate levels in the blood. So, that's all about folic acid vitamin. Thank you.